As we know, for T cell maturation and regarding T cell, uh, we have known that bone marrow produces all the cell lines, and T cell also came from bone marrow, and they are immature, and they need maturation in order to work properly. So for maturation, the T cell goes to the thymus, goes to thymus gland for maturation. And in the thymus, first they encounter the subcapsular zone. In here, they are immature. They are immature. They don't carry any receptor. So CD4 is negative, CD8 is negative, TCR T cell receptor is also negative. There is no any receptor on the T cell on the T cells, and CD3 is also negative. These all are the general or the main receptor present on the T cell. We will discuss in the next topic about the cell receptors and cell surface markers or cell surface receptor. First, in the subcapsular zone, they are immature, so they are not CD or not CD. No, they don't have any CD positive 8 or CD positive 4, and even don't have the TCR positive, so they are called double negative. So in order to gain these receptors to work properly, they goes to the cortex. And in the cortical epithelial cells, they undergo a process known as positive selection. In positive selection, the T cell express CD4 positive, CD8 positive, and both are positive, so this is double positive. And after this positive access, they undergo tcrDNA rearrangement, and they access TCR positivity. So now the cell is TCR positive, CD3, we can say CD3 positive, CD4 positive, and CD8 positive. Now the cell goes to the medulla section. First, why this double positive or this positive selection is necessary? So this positive selection makes the cell responsive for MHC antigen presentation. As we have known in our previous lecture about MHC, when the foreign antigen, either exogenous or endogenous, they came across the antigen presenting cells, the antigen presenting cell or MHC classes, they have high affiliation with the T cells, either CD4 positive and CD8 positive. So the TCR positive and the MHC affinity is very much important in undergo the T cell response. So if this response is not happen, then the body or the antigen presenting cell keep on alerting the body that there is a foreign antigen, do something. And if TCR is not responsive to that antigen or that MHC alerts, then the body is prone to infection, prone to autoimmunity, prone to cancer. And our T cells, we have T cell but we cannot use them, so the T cells are not functioning, so dysfunctional T cells. So that's why this positive selection is very much important, and it confirms that and it uh, we can say it control, or it monitor the cells, monitor cells which express first, which express TCR, the cells which express TCR, and they bind to MHC. Only those cells are survive. Rest of the remaining cell which are TCR, not expressing TCR, they undergo apoptosis. So this is the purpose of cortical epithelial positive selection. So when the cell has gone through the positive selection, then it goes to the medulla section, and it goes to the medullary, epithelial, and dendritic cells. So in the epithelial or dendritic cells, the medulla undergoes negative selection process, negative selection, and this negative selection is under surveillance of autoimmune regulator, AIR. This regulator monitor or surveil those cells, the cells, they monitor the cells having affinity low or high, high or low. The cells having high affinity for the antigens, self antigens. The cells having high affinity for self antigens are apoptosed, or they are converted into regulatory T cells. So only those cells are survive which has low affinity for the self antigen. Whenever the cells expressing TCR, CD8 and T4 they have high affinity for self antigen, then it relates to autoimmunity. For example, we have a dysfunctional or deficiency of autoimmune regulator. We have no regulator. Then the body is prone to autoimmunity. Like the cells are not functional, T cells are not functional, they are prone to autoimmunity. They detect self antigen as foreign and try to kill them. And this dysfunctional or deficiency of air, we have discussed it in the last immunodeficiency topic. So this air deficiency can cause chronic mucocutaneous candidiasis, which is a recurrent candidiasis, and its classic form is air deficiency, and this is mucocutaneous candidiasis. Also we have adrenal problem, adrenal insufficiency, the body cannot cope with any kind of stress, and then hypoparathyroidism, right? So this negative selection confirms that the cells having low affinity for the self antigen, so they can detect foreign antigen precisely. So apoptosis or T regulatory. For T regulatory, we will discuss this in next slide. For T regulatory cells, the main significance to converting the high affinity self antigens into the T regulatory cells, then they can suppress those uh, high affinity self antigen high affinity T cell and suppress their CD4 or CD8 receptor, so they are no longer causing autoimmunity, and the cell it can gain a kind of tolerance. So this is all about the T cell maturation, and next we will discuss how a T or B cell is activated.